Good morning, everyone. Today we are going to discuss about general pharmacology. So, first, what do we mean by pharmacology? The science of drugs. That is Greek word. Pharmakon means drug and logos it means study. So, the pharmacology is a study of therapeutic value and or potential toxicity of chemical agents on biological systems. In simple terms, it is the study of all the aspects of drug. It targets every aspect of mechanism for the chemical actions of both traditional and novel therapeutic agents. Two important and interrelated areas are pharmacodynamic and pharmacokinetics. Pharmacodynamics. In Greek word, the dynamic means power. Pharmacon means drug, we all know. What drug does with the body are the study of molecular, biochemical and physiological effects of drugs on cellular system and their mechanism of action. Then pharmacokinetics, it means kinesis means movement. So what the body does with the drug, that it deals with the absorption, distribution, metabolism and excretion of drugs. This comes under pharmacokinetics. So totally this pharmacodynamics and pharmacokinetics means the pharmacology. So here comes the diagrammatic representation of pharmacodynamics and kinetics. Pharmacodynamics means the action of a drug on the body. It may be receptor interactions, therapeutic actions, toxic actions such as adverse drug reaction, etc. And here comes the pharmacokinetics means the action of the body on the drug. It means absorption, distribution, metabolism, elimination. Pharmacotherapeutics. It is the application of pharmacological information together with knowledge of the disease for its prevention, mitigation, or cure. Selection of the most appropriate drug, dosage, and duration of treatment taking into account the stage of the disease and the specific features of the patient are a part of pharmacotherapeutics. Toxicology. Here in these slides we are discussing about the terms which are important in the pharmacology. So next comes the toxicology. It is a study of poisonous effects of drugs and other chemicals with emphasis on detection, prevention and treatment of poisonings. So these other chemicals include household, environmental pollutant, industrial, agriculture and homicidal. It also includes the study of adverse effects of drug, since the same substance can be a drug or a poison depending on the dose. So it is nothing but a drug which can act as a drug or a poison, because in additional doses or the doses which are above the therapeutic window will create some adverse effect, and that it may can become as a poison. That's what it means. And here, clinical pharmacology, it is the scientific study of drugs, both old and new in man. It includes pharmacodynamic and pharmacokinetic investigation in healthy volunteers as well as in patients. Evaluation of efficacy and safety of drugs and comparative trials with other forms of treatment, surveillance of patterns of drug use, adverse effects, etc., are also part of clinical pharmacology. The aim of clinical pharmacology is to generate data from or optimum use of drugs and the practice of evidence based medicine. This is known as clinical pharmacology. And next comes the chemotherapy. It is a treatment of systemic infection or malignancy with specific drugs that has selective toxicity for the infective organism or malignant cell with no or minimal effects of host cells. Drugs in general can thus be divided into pharmacodynamic agents. These are designed to have pharmacodynamic effects in the recipient. Then chemotherapeutic agent. These are designed to inhibit, or kill invading parasites or malignant cells but have no minimal or pharmacodynamic effects in the recipient. So this means nothing. Chemotherapy is a word which is commonly used for both the microorganism, for killing the microorganism, or for the malignant cells, that is tumor cells, cancer cells. So the both the substances, the chemicals which are used for killing the microorganism to prevent the infections, or to reduce or inhibit the growth, suppress the growth of cancer or tumor cells to prevent the cancer, that is known as chemotherapeutic agents. So this word will be applicable for both the conditions. And now the history of pharmacology. Knowledge of drugs and their uses in disease are as old as history of mankind. Primitive men gathered the knowledge of healing and medicines by observing the nature, noticing the animals, while ill and personal experience after consuming plants and herbs as remedies. So, in the primitive time, the men uses the natural sources such as for drugs. That what it means. And next point, ancient civilization discovered that extracts from plants, animals, and minerals had medicinal effects on body tissue. These discoveries became the foundation of pharmacology. It is an intellectual interest to know how drugs are discovered and developed. Often in the past, this was based on folklore or intelligent observation. Example, digitalis leaf, penicillin, etc. Nowadays, new drugs are mostly developed by the organic chemists working with the pharmacologists, increasingly from basic knowledge about key molecular targets. Usually, some sort of biological screen is used to select among organic molecules for optimum pharmacological activity. Historical developments in pharmacology. PNPSAO. It was a great herbal materia medica written in China. Then Kahun Papyrus is an oldest Egyptian document containing information about veterinary medicines and uterine disease of women. Ebers Papyrus, also an Egyptian document containing information about number of diseases and 829 prescription where castor oil, opium like drugs are being used. Hippocrates, a Greek physician, father of medicine known as first person who organized disease as abnormal reaction of the body, he introduced use of metallic salts for the treatment of disease. Theophrastus, a great philosopher called father of pharmacognosy, he classified medicinal plants on the basis of medicinal characteristics for the preparation of drugs. Dioscorides, a Greek, produced one of the first materia medica of approximately 500 plants and remedies. Claudius Galen first attempted to consider the theoretical background of pharmacology. Paracelsus, a Swiss scholar and alchemist, often considered grandfather of pharmacology, he introduces the use of chemicals for the treatment of disease. Valerius Scotus, he compiled the first pharmacopoeia. Francois Magendie, a French physiologist, laid down the dictum facts and facts alone are the base of sciences. Frederick Sertner, German pharmacist assistant, isolated morphine, the first pure drug in 1805. Claude Bernard investigated the plant extract curea and proposed the site of action of this agent, considered father of experimental medicine. Rudolf Bichheim, a German pharmacologist in 1847, established the first laboratory devoted to experimental pharmacology. Oswald Schimmelberg, father of pharmacology in 1872, set up an institute of pharmacology in Strasbourg, France. J. N. Langley and Sir Henry Dale pioneered pharmacology in England. John J. Abel established the first chair of pharmacology in the USA. L. Mayer Jones, regarded as father of modern veterinary pharmacology. Next comes the scope of pharmacology. It provides the rational basis for the therapeutic use of drug. So here, the pharmacology, by knowing the pharmacology, we can prescribe the rational use of medicine without producing any adverse effects or less adverse effect with low risk. So we can, that is the first point. Then before the establishment of this discipline, even though many remedies were used, but doctors were reluctant to apply scientific principles to therapeutics. In 1920s, many synthetic chemicals were first introduced and the modern pharmaceutical companies began to develop. The Second World War was the impetus for accelerated research in pharmacology in the US and introduced strong analytical and synthetic chemical approaches. Scientific understanding of drugs enables us to predict the pharmacological effect of a new chemical that will produce a specified therapeutic effect. The scope of pharmacology has expanded greatly over the last decade to incorporate many new approaches such as computer assisted drug design, genetic screens, protein engineering, and use of novel drug delivery vehicles, including viruses and artificial cells. These are all the scope of pharmacology. Then we can go into the definition, not definition, meaning of drug. In French word, drug means drug, a dry herb. It is a single active chemical entity present in a medicine that is used for diagnosis, prevention, treatment, and cure of a disease. WHO states that any substance or product that is used or intended to be used to modify or explore the physiological system or pathological state for the benefit of the recipient. Nature of drugs. All drugs are chemical entities with simple or complex molecules. Majority organic compounds. Weakly acidic, weakly basic, are non-electrolytes. Means weakly acidic example aspirin or penicillin, weakly basic morphine and chloroquine, and non-electrolytes alcohol or diethyl ether. Most are solids, example paracetamol, propranolol, furosemide, ampicillin, etc. Some liquids, ethanol, glyceryl trinitrate, propofol, castor oil, and few gaseous, that is nitrous oxide. Some purely inorganic, lithium carbonate, ferrous sulfate, magnesium hydroxide, etc. So these are the nature of drugs. Then molecular weight, majority of drugs range of 100 to 1000 Dalton. Molecules less than 100 Dalton, no sufficiently specific features, shape, size, configuration, chirality, distribution of charges, etc. to selectively bind to only one or few closely related target biomolecules to the exclusion of others. Larger molecules than 1000 Dalton do not readily pass through membrane or barriers in the body to reach the target sites in various tissues or cells. Therefore, the majority of the drug ranges from 100 to 1000, not less than 100 or not more than 1000 Dalton. That is the optimum. And next few drugs are small as lithium ion 7 Dalton, and some like heparin 10 to 20 kD, gonadotrophins more than 30 kD, enzymes, proteins, antibodies more than 50 kD are much bigger. Bulky molecules drugs have to be administered parenterally. Drugs, xenobiotics, many endogenous chemicals, hormones, autocoids, metabolites, or nutrients are also used as drugs. That is, xenobiotics means nothing, it is a chemical substances. Plant sources, alkaloids, atropine, morphine. Atropine is extracted from Atropa belladonna, morphine from Papaver somniferum family, and glycosides, example digoxin from Digitalis purpura, oils, essential oil, fixed oil, etc. Essential oil, volatile oil, it is also known as from leaves and flowers of clove, peppermint, eucalyptus, etc. And fixed oil seeds, example groundnut, coconut, castor, olive oil, etc. Mineral oil, example liquid paraffin, hard and soft paraffin. Then other substances are gums, resins, tannins. Gums means excretory products, gum, acacia. Resins, tolu balsam, cough mix, which is used in cough mix. Then tannins, catechu. Then second comes the animal sources. From animal sources, which are the drugs or chemicals that can be used are hormones, vaccines, sera, vitamins. The examples of hormones are insulin, that is from pork, porcine, beef, bovine, etc. Then vaccines are polio, anti-rabies. So next one is vaccines, that are the examples for polio and anti-rabies. And then one is sera, ATS, anti-tetanus serum. Then vitamins, vitamin B12 from liver extract. Then microorganism, the examples from drugs used here, antibiotics, minerals, iron salts, calcium salts, lithium carbonate, magnesium, aluminum hydroxide, iodine, etc. Then synthetic chemicals, synthetic glucocorticoids, benzodiazepines, thiazides, etc. Recombinant DNA tech, that is human insulin, HGH, interferons, erythropoietin, etc. This is the recombinant DNA technology. This is a diagrammatic representation of recombinant DNA technology. From human DNA, the gene of interest will be separated and it is inserted into the bacterial cell, that is known as transformation, and bacteria with gene of interest is formed. Then another from plasmid, that is cloning vector, cut with the restriction enzyme, and that part is used, that is recombinant plasmid, and it is again incorporated into the bacteria with gene of interest, and it is produced. That is known as recombinant DNA technology. Thank you. That's all for today's session.